everyone. Currently, we are discussing about our immune system in this module. And during the last two lectures, we discuss about our innate as well as adaptive immune response. Almost in a storytelling way, I presented that how our immune system protect us while we are getting infected by some pathogen. It could be bacteria or it could be virus, or anything else also, but our immune system very precisely and cleverly try to prevent that infection and protect us. Today, particularly in this lecture, I will be discussing a little bit about mechanistic understanding, how some of those things are happening. Particularly, I will focus on understanding immunoglobulin fold present in T cell receptor, B cell receptor and antibody. And also I will be discussing about the structure-function relation of an antibody. As you already know that antibody, they are very precise weapon of our adaptive immune system, and it specifically target specific pathogen, right? So I will be discussing about their structure-function relation, and finally the molecular basis of diverse antigen specificity. So let's start. Here in this panel here, this is our TCR, or I would say T cell receptor. And as you can see, this is the membrane, right? Transmembrane region, that's why we are mentioning. And on the outer side of the cell, this is T cell, right? This is T cell. So you have this T cell receptor, and this is particularly, this TCR is called alpha beta TCR, because as you can see, it has one alpha chain and one beta chain. But one of the most interesting thing here is whether we are talking about TCR or BCR or an antibody, you will be seeing that those proteins are made up of an unit. It is called immunoglobulin fold. Okay, immunoglobulin fold. So immunoglobulin is the other name of antibody. Since the same fold is also present in antibody, and this overall structural fold we mentioned as immunoglobulin fold, or simply IG fold. So as a result of that, if you see, so we have one IG fold here, another IG fold here, here and here. So all are IG fold. They have somehow some kind of structural similarity. And since those are membrane protein, sometimes some carbohydrate group, they are also attached with this protein, but for the time being we can ignore that. So we are mostly discussing about immunoglobulin fold, antibody structure and their antigen recognition. Now if we concentrate on this panel here, so this is our BCR, that means B cell receptor. Also it is antibody. This is kind of very similar. As I already mentioned before, that BCR means they are attached to the cell surface, they are attached to the membrane of B cell, and antibody, they are the secreted form of the same thing. So as a result of that, while we are discussing their structural organization, the BCR and the antibody, they will be kind of similar, same. So here, as you can see, you have multiple immunoglobulin fold present in this antibody, right? So how many? Particularly, this is a specific type of antibody. This is called IgG. There are, different classes of antibody. I will discuss little later. So here, if you see, multiple immunoglobulin fold there. So here in this case, 12 Ig fold present, 12 Ig fold. And as you can see, this is, as I'm just drawing here, this is one polypeptide chain, this is another polypeptide chain. So this polypeptide chain is bigger and its molecular weight is more, and this chain is called heavy chain. Heavy chain of an antibody. On the other hand, this one is the smaller one, right? Smaller chain or the lighter chain. This is called light chain of the antibody. Now, as you can see, this heavy chain and light chain, they are attached by some bond. This is disulfide bond, okay? And also this heavy chain, they are also attached together by disulfide bond. So now, if I, discuss this, for example, this is made up of 4 polypeptides basically, 2 heavy chain and 2 light chain. So as a result of that, there are different biophysical and biochemical techniques. For example, one of those is SDS page, right? SDS page. So this is some kind of gel electrophoresis technique where we analyze proteins, particularly their molecular weight. So as a result of that, if we run an SDS page and we load antibody, so then how many bands we will get? So we will get 2 bands, because this is SDS page. It is, like it will denature the protein, and in SDS page generally we add some reducing agent, either beta-mercaptoethanol or DTT, dithioethitol. So they will reduce the disulfide bond. So as a result of that, you will get 2 bands. But now we have 4 polypeptide chain, but we are getting 2 bands because these 2 light chain, they are identical, as well as 2 heavy chain, they are also identical. And also I must mention that this antibody, they will recognize antigen. How many antigen? 2 antigens. So here 1 antigen and here another antigen. So here antigen will be recognized. But this antigen, say for example antigen 1 and this antigen, another antigen, right? But the same antigen, it should be also antigen 1. Why? Because they are identical. Their antigen binding site here is identical. So as a result of that, they will see the same antigen by the 2 arm. And I would also like to mention here that this Ig fold, that every unit here, those are Ig fold. As I mentioned that 12 Ig fold, you can now understand from the figure why 12 there. And here the terminal region here, which is capturing the antigen, this is called the variable domain. So here mentioned that this nomenclature is very important, because in the future slide also you will be seeing kind of similar thing. So see VL, that means the variable domain of the light chain, VL. So this way we are mentioning VL. Similarly here VH, the variable region of heavy chain, VH. Then here CL, constant region of light chain. And now here CH1, CH2 and CH3, constant region of light heavy chain 1, 2 and 3. So this is the antibody structure. And here this portion is also very important. Although I mentioned that the variable region, they are important to capture antigen, but through the constant region they carry out some effector function. Effector means, so this portion is called the FC region. FC region. So FC stands for here fragment crystallizable region. Scientists observe that this fragment, if we can make this fragment it will be very easily crystallized. So fragment crystallizable region. So these bind to some cell surface receptor and then recruits other cells to carry out some effector function, so that the immune cells can clear the infection in a better way. So as a result of that, although they are not directly recognizing the antigen, but they mediate cell-based function. And that is also important. I will be discussing in detail. Now, the structural features of immunoglobulin fold. Whatever I mentioned about the BCR or antibody structure as well as the TCR structure, their units are immunoglobulin fold. And it is a protein. And if I start from this N-terminal end, as you can see that this protein is made up of beta strand. During the protein lecture, you have, you came to know that protein, they have alpha helix and beta strands, all those things. So here, as you can see, this is all beta strands. And this is the N-terminal region, and this is the C-terminal, the end of the protein. And on top of that, I must say that beta strand, but what kind of beta strand? This is all are anti-parallel beta strand. Anti-parallel beta strand. Now, this is just the topology diagram. What is present in this protein that we are trying to show. But now, one important thing is this protein, this immunoglobulin fold, they have some conserved thing. So between B and F strand, so all these beta strand, we just put some name, some convention is there. I am not discussing that. But in between B and F strand, they have some conserved disulfide bond. And this is present in all immunoglobulin fold. And now, just we are presenting this diagram in one plane, right? You just imagine that on your paper or notebook, it's present like this. Now, if I draw an imaginary axis here, and then it can be fold. So the idea is, I would say, if I see this is just in one plane, all these beta strands are showing like this. But now, finally, it will fold like this. So from here to here. So this is our IG fold. So that way, if I fold, then it will give rise to this kind of structure. Okay. So that's why I colored in a different way, that G, F, C, C prime and C double prime, they are present on one face, and the other strand A, B, E, D, they are present on the other face. So something like this. And now this is called beta sandwich topology of immunoglobulin fold. This is kind of beta strand, and what kind of thing? This is sandwich. When we make sandwich, for example, two bread, we are putting like this. So that's a beta sandwich topology. And inside we have multiple hydrophobic residue also that kind of stabilize the structure. In addition to that, this conserved disulfide bond that also stitches these two beta set together. So this is the common structural motif of immunoglobulin fold, and this is very important to understand so that I can discuss the later part of this of this discussion. So now, if you see the mechanism of antigen recognition by antibody. So in the previous diagram, I mentioned that 12 immunoglobulin fold is present, but this is real structure. So this is some kind of three-dimensional structure. So this is some coming from some X-ray crystallographic structure. As you can see, so this is that VL, that means the variable domain of the light chain, and this is VH, the variable domain of heavy chain. Whatever it is, but this portion is the antigen binding site. And now the question is how it captures the antigen. So as a result of that, now if I am showing the topology diagram of either this VH or VL, then you will be able to understand, right? Because VH and VL together, they capture the antigen. So as a result of that, this is the topology diagram, whatever I presented there, but here I just made by the same color here. And here 3 loops, they are very important. So as you can see, this is the N terminus and C terminus. So these 3 loops, 1, 2 and 3, those 3 loops, they are called CDR loops. CDR stands for complementarity determining region. Okay, complementarity determining region, because they are complementary to the antigen, so they bind to the antigen directly, right? And they are also sometimes called that HV loop. HV stands for hyper variable. So here you can see 1, 2 and 3 loops present here. This is called CDR1, CDR2 and CDR3. They are capturing the antigen. That means total 6 loops they are working together. Why 6 loops? Because 3 loops from this VH and 3 loops from this VL, right? So total 6 loops, they are working together to fit with the antigen. Now the question is, I mentioned that antibody can be of countless different types. They can capture different types of antigens. So as a result of that, their features in this loop should be different, right? So now here some interesting thing is coming, because as you know, we have approximately 20,000 protein coding genes. But we are saying that we are getting countless antibody throughout our lifespan. So as a result of that, they are not the reflection of the direct genetic, direct product coming from the, DNA. So that not like, the correlation is something different. How we are getting so many different types of product, right? If it's directly getting encoded from the, whatever present in our DNA. So the reason is that some more complicated thing is going on here. Particularly, I would like to mention in case of TCR, BCR and antibody, in all those cases, like a special thing is going on, some kind of recombination is going on, which I will be discussing, and that is giving you different types of different combination of this loop and this variable region of antibody or BCR or TCR, and that help them to capture different types of antigen. Now here this is one interesting thing. This is whatever I mentioned here, this is conventional antibody, where you have both light chain and heavy chain. But this is some kind of unconventional antibody. This is found in camelid group of animal. So camelid means, for example, camel, alpaca. So this is a little smaller animal than camel. So this camel, alpaca, llama, so those animal, they have this kind of antibody. This is called heavy chain only antibody. Heavy chain only antibody. Why? Because as you can see, they don't have light chain. So somehow they evolve in such a way that this is good enough to capture antigen, right? So antigen binding site. So similarly here another antigen binding site. So as a result of that, like this kind of unique antibody, it has some relevance in terms of therapeutics. Okay, so in right time I will discuss that. Now here antibody structure are like early days when scientists were kind of trying to determine antibody structures and its different types of biophysical, biochemical properties. They found that this antibody, when we are treating this antibody with some enzyme, particularly proteolytic enzyme, pepsin, okay. So this pepsin will digest this antibody at several places, and you will get this fragment, a whole fragment here. So this region is called the hinge region, okay, hinge region of an antibody. So just below the hinge region. So as a result of that, you will get this segment, and here small pieces from the Fc region also coming out. So this segment, if you see, they have the antigen binding capability. So this FAB, particularly if you see it's mentioned FAB, this means fragment antigen binding domain. Fragment antigen binding, FAB, fragment antigen binding domain. And FAB hold 2 because after pepsin digestion, as you can see, these two FAB fragments, they are staying together. On the other hand, if you digest the same antibody with another enzyme called papain, so this is also a proteolytic enzyme, and it will cleave above the hinge region. So as a result of that, you are getting two FAB region here, fragment antigen binding region, and this Fc region, as I already mentioned, fragment crystallizable region. Correct? So that way, antibody was characterized in much detail. And here I would like to mention not only this FAB region. So say FAB region, they have still the capability to capture the antigen because they have the antigen binding site here. And because of the genetic engineering approach, we can do many things through genetic engineering approach. This is the conventional antibody. We are just treating with some enzyme, proteolytic enzyme. But it is possible to make this kind of fragment. Only the variable region of the heavy chain and light chains are present. This is called FB, the fragment variable region. Okay, fragment variable region. It will also bind to the antigen. Only thing, these two variable domain of this light chain and heavy chain, they might fall apart. So as a result of that, in order to do this kind of construct FB, fragment variable region, we have to stitch these two domains together by some amino acid linker. That's all. And this is our unconventional antibody, right? Heavy chain antibody. No light chain present here. So interestingly, from here you can get this fragment. You can generate through genetic engineering approach this fragment. This is just the VH domain, the variable domain of the heavy chain, but still it can capture the antigen. Why I am telling you? Because antibodies are now being used for different immunotherapeutic purposes, and sometimes we try to make antibody in a very small size, because this conventional antibody is really big. Overall molecular weight should be approximately 150 kilodalton. Okay, but now for different purpose, clinical purpose, if you want to design smaller antibody, so one could be possible like FAB fragment, another one is FB fragment, and this is the smallest fragment possible. Sometimes this is also called nanobody, and it has therapeutic application. Now, I would like to mention a little bit about the recognition of antigen by T cell receptor, because I already mentioned that how CDR1, CDR2 and CDR3 of the variable region of heavy chain and light chain, they capture antigen, and I particularly mention because of some some kind of recombination, this sequence, the structure of this variable region, they alter a lot, and they can capture different types of, countless different types of antigen as well. Now let's see what is happening in T cell receptor. So the basic difference here between the antigen recognition by T cell receptor and the B cell receptor or antibody is that TCR recognize processed antigen, only kind of small peptide which is presented by MHC molecule or major histocompatibility complex. For example, this is MHC class 1. This is the membrane. So on the antigen presenting cell, this class 1 MHC may be presenting some peptide here. Okay. So this is our peptide now. So it looks something like this. This is the three-dimensional structure from the top view. If you see from the top, you will be seeing that this is our peptide here. Now, B cell receptor, they can directly bind to the antigen. They don't need any MHC mediated presentation. They can directly bind to the antigen of different size as well. But in this case, peptide is being presented by the MHC, and now look how they bind to the TCR. So this is on the top side, I am showing you the TCR, T cell receptor, and this is alpha beta TCR. As you can see that alpha and beta chains present, and this is the variable region of the beta chain, and this is the variable region of alpha chain. Just like the variable region of antibody, here also you will be able to see that you have 3 complementary determining region, the 3 hypervariable loop present. Similarly here also 3 loop present. So these loops, they bind to the peptide which is being presented by the MHC. Here this thick yellow color diagram, this structure, this is the peptide, the antigenic peptide which is being presented by this MHC molecule. Now, as you can see, this red portion, this is little different. This is hypervariable loop 4. This is, this help the TCR to bind to the MHC itself, because we have to understand in this case, TCR and MHC, they have some kind of relation. So the TCR will see some antigen loaded on a specific type of MHC. So not just the peptide, related to some relation with the MHC as well. So this is called the MHC restriction. So little bit more complex, but the overall, like I would like to say that TCR, they can also generate variation in their variable region, so that they can recognize different type of antigen which are being presented by MHC molecule. Now, the molecular basis. Why TCR, BCR and the antibody, they can see different types of antigen. How they are changing their structural features, the binding properties. This slide is little complicated, although this is very like I would say it's a very common thing, but it's complicated. That sometimes we say that VDJ recombination. This is the relation. You will understand that why we are getting so many different types of antibody. So in this case, as you can see, although very complicated slide, but I will go step by step. So just now for the time being, think about this is, we are discussing about antibody. But this is same with the TCR also, it happens in the same way. So this is the light chain and this is the heavy chain of antibody we are showing here. So in the light chain, this is the DNA level we are showing, the germ-like germline DNA. This is the variable region segment, this is the joining region segment, and here another one here present, this is the constant region of the light chain. Okay. So now, in the same way, if we say about the heavy chain, you will get the variable region segment and the joining region segment, but here something extra here. So D stands for here diversity, and J stands for here joining region, V stands for variable region, and C they refer to the constant region of the antibody. Now, what is happening? They are undergoing some kind of recombination that is called somatic recombination. So as a result of that, in case of light chain, this V and J, when they join together, some kind of shuffling happen. And here you can see that V and J, they are joined together. And here constant domain is also coming. Then what will happen? After this rearrangement of this DNA, then because of the transcription, you will get the primary transcript. So this is the RNA now. Since it is eukaryotic RNA, it will have the poly A tail we are showing here. Then what will happen? In case of eukaryotic RNA, splicing will happen. So as a result of that, for example, this portion is going away. So after splicing, this is our mRNA, the mature mRNA here. So as you can see, V, J and C, they are continuously present. And after translation, you will get, this is translation step, you will get the polypeptide chain. What kind of polypeptide chain? The light chain of an antibody. Now this L stands for here, L equal to leader sequence. Why leader sequence? Because all this transcription and then translation, or translation particularly I would say, that is happening inside the cell, in the cytosol. But this protein, they should be on the surface of the cell or they should be secreted. So they need some kind of signal so that they will come out from the cell. They will be either on the cell surface or they will be totally released, but they need this leader sequence, that's all. But that will not generate any diversity among the antibody. But anyway, here major thing is that the recombination, the somatic recombination here between V and J region in case of light chain. And now in case of heavy chain, whatever I mentioned that remains same. In addition to that, we have one extra D region, the diversity region. That's why this is called VDJ recombination, and sometimes we put in this way the D inside the bracket because the D region is not present in the gene corresponding to the light chain. That is the reason. So as a result of that, from here you will get this full heavy chain, and on the left panel you are getting the light chain. And this portion, the variable domain, this is the variable domain of heavy chain and this is the variable domain of light chain. That is constantly shuffling, and the correct one which can hold antigen, which can bind to antigen in a better way, so that will be selected, and that's why we are getting different types of antibody structure and they are helping us. Clear? So this is called VDJ recombination, and it generates the diversity of antibody as well as TCR and BCR. Now, in addition to this VDJ recombination, whatever I mentioned, we have some something extra as well. That is particularly for antibody, not for TCR. Okay? So what is happening? Beside VDJ recombination, another important thing is happening that is called somatic hypermutation. So now thing is, like at the DNA level, I told that that rearrangement is going on, right? But those are still either on TCR or on BCR. So BCR, they are present on the surface of B cell, but they are not yet plasma cell. But plasma cell will secrete antibody with very high affinity. They can bind to antigen with very high affinity. So during that time, that the plasmocyte transformation, like the plasma cell particularly, I would say. So during that time, another thing happened that is called somatic hypermutation. So after that recombination, like some kind of mutation happened, and hypermutation, lot of different types of mutation, random mutation happened in the gene which produce this antibody, not TCR anymore, antibody. So as a result of that, they shuffle different types of antibody and they select the best antibody which can bind very tightly to the antigen. So as a result of that, some kind of affinity increased. So this is called affinity maturation of an antibody. And after affinity maturation steps, then that B cell now will be converted into plasma cell, and it will secrete antibody, and this will bind to the antigen very tightly. And besides somatic mutation, another important thing is there, that is called class switching. Although whatever antibodies mentioned, that conventional antibody I referred, those are IgG. But there are other group of antibody as well. For example, IgA, IgD, IgM, and IgE. So now, if you see like antibody structure, say something like this, like this is heavy chain and this is light chain. And I mentioned this is the Fc region. Now, this Fc region, that can also be switched between different classes of antibody. That means the antigen binding site remains same, but their Fc region can be changed. So that is, I'm not going into details, molecular details, but that step is called class switching, and that will bring different type of, I would say, diverse type of effector function. So effector function means when this antibody will bind to antigen. For example, like that antibody is against some bacterial protein which is present on the surface of bacteria. So this is one bacteria, and this antibody actually raised against some protein present on the surface. So what we expect? So the antibody, after secretion from the plasma cell, they are binding to the antigen present on the surface of bacteria. Now the Fc region, so this is the Fc region, okay? Fc region, some immune cells, they have the Fc receptor and different types of Fc receptor present. For example, Fc receptor like Fc gamma, Fc epsilon, depending on different types of immune cell. So now this Fc region will bind to that Fc receptor present on those immune cells, and they will recruit other immune cell so that they can kill this pathogen very effectively. They can clear the infection very effectively. This is effector function. For example, this Fc region can bind to the Fc receptor present on NK cell, okay? It can present on mast cell. This is also another type of immune cell. It can bind to DC, macrophage. So now all those cells, like they will create or they will bring the cell mediated immunity because of this effector function. So this is, I would say, kind of idea about how antibody that helps in the clearing infection, right? It's very powerful tool although, and particularly this VDJ recombination, somatic hypermutation, and class switching, they are very important for the production of this antibody. And here I would like to mention here, this is little bit of philosophical idea. If you see, particularly this B cell, their lifestyle is, they are part of very advanced or developed, immune system, right? In mammals, like we have this kind of complex scenario. In some other vertebrates also they have immune system, but I'm like mostly concentrating on mammalian immune system, human immune system. But as you know that, in case of microbes, sometimes they acquire some genetic character by horizontal gene transfer, right? As I mentioned in the previous module. But here, the horizontal gene transfer is not happening. But what is happening? Some kind of, shuffling of genetic information, and that is giving them kind of additional property. They can evolve themselves to make different types of antibody. And like prokaryotic cells, so they are not also bound to a particular tissue. They are flowing through blood. They are flowing to lymphatic vessel, lymphatic duct. So as a result of that, some kind of analogy there. But that's okay. Now it is fine. So this way I would say T cell recognize antigen because of this VDJ recombination, because of the variability in TCR in that variable region, and particularly B cell, they produce BCR as well as antibody, and that's all.